Hey everyone. Okay, so I'm in the I'm in the rainforest, living in a little house with like seven other people. So if you hear any weird sounds, I don't know. That explains it. Let's talk about oncology. So specifically with an emphasis on leukemias, lymphomas, things like that. All right, so we're just going to do a little introduction to cancer, and then we're going to talk about just an overview of all of the specific proliferative disorders, emphasizing the myeloproliferative disorders and the plasma cell dyscrasias, then the leukemias, lymphomas, and then a final video with the chemotherapeutic agents. Some cancer basics. So the general outline of this first video, this first chapter, just a little some nuts and bolts of cancer, the 10 hallmarks, which is based off of this famous paper we'll talk about. A lot of nomenclature, different types of carcinomas versus sarcomas, specifically cancer-causing agents, whether this be genes, carcinogens, or different pathogens, and then specifics about staging, tumor markers, different stains we can do, and then we're going to wrap up with a little bit on paraneoplastic syndromes. All right, so cancer takes a long time to develop, usually, right? Some cancers might develop really quickly in a year, some might take 20 years, but on average it takes a couple of years. So, you have a normal cell, is a genetic change, and then maybe it divides a little bit more than it used to. Another genetic change, and now maybe it doesn't proofread the DNA as well as it used to. Another genetic change, maybe now it can break through the basement membrane, and et cetera, and then maybe a final genetic change to allow it to invade into a new foreign region, and then we might say that it's a malignant tumor. So it takes a long time and a lot of gene mutations typically. So at least a couple. We might say about 6 on average, but several changes takes a long time. Specifically, signs of cancer: fever, night sweats. Cancers secrete pyrogens. Weight loss, also known as cachexia, if it's extreme. Cancer cells divide a lot, right? That's the whole point of cancer, is that it just divides uncontrollably. This takes a lot of energy to replicate the DNA and everything inside of the cell. They're also really inefficient with their energy. So the Warburg effect refers to the idea that cancer cells don't really do aerobic respiration. They just do glycolysis. If you recall, we get a measly 2 ATP with a glycolysis, and we get something like 30 to 32 ATP when we do aerobic respiration. So they're crazy inefficient on top of needing a lot of energy, meaning that they're going to divert a lot of the resources that go towards you towards the cancer. And so you might become frail and the cancer enlarges itself. Additionally, the TNF, tumor necrosis factor alpha, induces catabolic activity in nearby areas. So this is kind of an interesting, complex relationship between the immune system and TNF and cancers. So on the one hand, TNF is supposed to be, in other inflammatory mechanisms are anti-cancer, but weirdly they're also utilized and co-opted by the cancer to facilitate cancer growth. And this is very complex and beyond the scope. But in this case, the TNF is facilitating and helping break down nearby tissue to feed the cancer. So the hallmarks of cancer was the seminal paper by Douglas Hanahan and Robert Weinberg in 2000. Just to show you how important it was, it's got 37,800 citations. Every 11 years, they basically done a revision to this paper. So 2011 was the newest paper, 57,800 citations, and they just came out with a new one. Only 6 citations, but it was literally just published, so probably not a lot of people have had a chance to subsequently cite it in their particular paper. So our understanding of cancer is constantly progressing. We're really going to be emphasizing the 2011, which has the 10 hallmarks, but so 2000 paper was the original 6 hallmarks. I highly recommend it if anybody is interested at all in oncology to read these papers. They're fantastic. 2011, they added 4 more. And then in 22, they've added another 4. So there's 14 now, but you know what? For your test, for your MBME, you just need to be familiar with these 10. So sustaining proliferative signaling just means oncogenes. Oncogenes turn on, pushing the cell cycle forward. Things like MYC, RAS, cyclin, CDK, receptor tyrosine kinases, these get upregulated. Next, we have basically inhibiting tumor suppressors, right? The cell cycle breaks, like the retinoblastoma protein, and P16, things like that. These are going to be downregulated and inhibited. Avoiding the immune system, so CTLA and PDL, which we will talk about later. So we can induce CTLA on T cells, or the cancer cells themselves can upregulate PD for programmed death ligand, which can turn off the immune system. Most cells have something called a Hayflick limit, so their telomeres get shorter and shorter and shorter. This kind of acts like a fuse for a bomb, which will eventually cause the cell to self-destruct. So maybe a cell has 50 divisions or so, and then it's no mas. But the cancer cells figure out a way to elongate the telomeres. Typically, this is by upregulating telomerase. So the telomeres don't actually get shorter, and therefore they can replicate as long as they want. So we just talked a little bit about the TNF secretion. They can either secrete TNF themselves, or more likely they will go ahead and manipulate nearby immune cells to be part of the tumor microenvironment to secrete tumor necrosis factor. The loss of cadherins is going to loose intercellular junctions so that the cancer cells don't necessarily have to be so close together. And on top of that, the metalloprotease is going to help them break through the extracellular matrix, break through the basement membrane. This is going to be necessary to get out of the local area into the lymph, into the blood, and into the new, the new organ that they're invading. Angiogenesis tumors, they need a lot of resources, so they need a lot of blood coming towards them. This is really just VEGF. So cancers like to have mutations. Mutations helps them adapt, helps them evolve. Things like BRCA for double-strand breaks, and MSH for this would be specifically for I believe this is microsatellites. I could be wrong, but microsatellites and just missense mutations that are recognized. These will get mutated so that the cancer cells can just develop more mutations. Apoptosis. So this kind of goes along with this over here. They're going to, so not just telomeres that will indicate when a cell should commit suicide, many different factors. And BCL2 is an anti-apoptotic protein. BCL2 actually stands for B-cell lymphoma. And we'll see this later. I believe it's follicular lymphoma and diffuse large B-cell lymphoma. And then they're going to downregulate P53. So P53 is pro-apoptosis, BCL2 is anti-apoptosis, and so both of these fall under this category. And then a bunch of things. So we talked about the Warburg effect a second ago, the idea that cancer cells are largely just going to do glycolysis. A bunch of different glycolytic enzymes and Krebs cell and electron transport things. An example being pyruvate dehydrogenase. You can just imagine that if this was turned off, you couldn't transform your pyruvate into acetyl-CoA and feed it into the Krebs cycle, so you would just have to do glycolysis. This is just an example. Many different enzymes are going to be a factor here. So a bunch of definitions to talk about. So dysplasia. Dysplasia is specifically talking about the loss of cell shape and size, so the loss of uniformity, and the idea that the nuclear to cytoplasm ratio is going to increase. And this is because they're dividing so quickly that they can't actually enlarge their cytoplasmic contents to normal values. So we see this a little bit here. Carcinoma in situ is just, more irregularity, and specifically it spans the entire epithelium, breaking through the basement membrane. So, we talked about metalloproteases, collagenases, angiogenesis to help, blood vessels sort of grow towards them so that they can have a path into the lymph and the blood, and not only, of course, just to feed them. So this is just going to be an invasive carcinoma. And then lastly, metastasis, which is where they go to a foreign area, foreign organ, either through the blood or the lymph. This is what it would look like on an actual histology. Normal, here we have a little bit of dysplasia. You can notice that we're seeing larger and more nuclei, and it's becoming slightly irregular as well. And then, it's just getting a little bit worse. And then here, this is going the entire length of the epithelium, but it's not breaking through. And so therefore, it is still in situ, or in situ. Neoplasm is just an abnormal growth of cells. Within this, we have tumor. So tumor is specifically talking about the large, swelling, gross morphological appearance. The big old bump. That's the tumor. And then a cancer is specifically a tumor that is malignant, that can invade foreign areas. So terminology is a little confusing, really important to get down. Different types of tumors. We have benign tumors, and then we have malignant tumors. So a couple differences. Benign are the good ones, malignant are the bad ones. Again, so slow and fast. These are going to be local. These can spread. This is going to be good. This is poor prognosis. Well demarcated, well differentiated. Well differentiated just means that they look like normal cells. The normal cells are differentiated. And for the malignant ones, these are not circumscribed and they are not differentiated. And, we can kind of see that here. Just to give you an idea, if you had, let's say, a mole or something, if it's symmetrical, if the borders are even, if it's just one color, if it's small, this is just a normal benign mole. But if it's malignant, then it's going to be asymmetrical, it's going to be uneven, there's going to be lots of colors, and it's going to be large. And that's a little bit what we were seeing here. So benign tumors are going to grow uniformly, and they're not as, if you have a mole, it's not necessarily something you should worry about. But a benign tumor does have the ability to transform into a malignant tumor. Two major types of cancers are carcinomas and sarcomas. Sarcomas are from the epithelium, so this is an epithelial cell lining our blood vessel, and this is our mesenchymal tissue, the connective tissue, the bones, muscles, fats, etc. The carcinoma is going to be the epithelial tissue dividing extra. And you can kind of see this here, that we're getting extra epithelial tissues growing there. And then the sarcoma being the myoepithelium, which is the mesenchymal cells, the bone cells, the muscle cells, etc. Here we can see that this is growing a little bit out of control. So mesenchymal cells are really just cartilage producing, bone producing, tendon producing, muscle producing. It could make fibroblasts, etc. There's the connective tissue, the mesoderm. That is going to make the sarcoma. So a little bit more about this. So epithelium, mesenchyme, older, younger. Most importantly is that the carcinoma goes through the lymphatic system. The sarcoma goes through the circulatory system. These are a better prognosis. These are a worse prognosis. This is less vascular, more vascular. Carcinomas you see a lot. Sarcomas you still see a lot, but I guess slightly smaller amount. So this is a lot. Just to break down a little bit more easily. So glandular, addin, squamous, not too hard. Bone, osteo, blood, heme if it's benign, angio if it's malignant, lymph, leiomyoma. So myo is muscle, so lyo is smooth. Rhabdo is skeletal. Myo is muscle. Cartilage, chondro, fat, lipo, connective tissue, fibro. So that's not too tricky. The benign is just going to be an oma, and then the malignant, if it's epithelial origin, this is going to be a carcinoma. If it's a mesenchymal, it's going to be a sarcoma. So we can just kind of throw all these together, right? If it's a bone, it's going to be an osteo. If it's a benign bone tumor, it's an osteoma. If it's a malignant bone tumor, it's going to be an osteosarcoma. So this is going to serve you guys not just for this exam, but for the rest of preclinical. Cancers show up all over the place, and it's helpful to know which ones are the bad ones, which ones are the easy ones. And this is just a good rule of thumb to get all of this down. All right, incidence. So the prostate and the breast cancers are going to be the most common for adults. And that's because these organs are very responsive to the endocrine system and various hormones, and the hormones will stimulate growth, and the growth is associated with cancer. So prostate for men, breast for women, these are the most common cancers, and therefore they're going to be also high up for the mortality. But it actually turns out that the lung is just the worst one you could have. So lung is just the worst cancer no matter what, no matter whether you're a man or a woman. Lung cancer is the highest mortality rate. But notice it is followed by the prostate and breast. So this makes it a little bit easier to memorize. Also know that for kids, leukemia, specifically ALL, acute lymphoblastic leukemia, this is the worst. This is the worst and the most common cancer. So again, not the most common leukemia, the worst common cancer, period, for kids. Cancer is the second leading cause of death. Cardiovascular, heart attacks, things like that. Strokes are the number one. A metastatic thing is a cancer that grows somewhere else, and then it travels to a new region and spreads its roots there and then grows again. And a primary one is just one that originates in that particular site. You can tell these just on a growth morphological analysis. A primary tumor is usually just going to be contained in one site, while when something metastasizes, some will go there and grow, some will go there and grow, some will go there, kind of like a weed. It'll just go and grow in many different sites. So you should be able to do, if they show this to you on the NBME, just be able to, boom, snap your fingers and you know which one is which. So we said carcinomas go through the lymph, sarcomas go through the blood. The lungs, so when you think about the lungs go to the pulmonary vein is returning to the heart, left atrium, left ventricle, it pumps immediately through the aorta. One of the first places it goes is just directly to the brain. So the brain has a direct flow from the lungs doesn't really have many other stops. It's unlikely you're going to have the cancer go in the heart. So kind of makes sense that that would be a common metastatic site. The colorectal, things in your GI system usually go through the liver for detoxification and processing. So this is also direct flow. So this one also seems kind of reasonable that a colorectal cancer would go to the liver. Prostate and breast both go to the bone. This one's a little random, but important to know. Axial, so it's going to be the spine and the skull, spine especially, and maybe the ribs. If you have a bone cancer, it's much more likely that it was a metastasis than it was a primary bone tumor. Example of a primary bone tumor might be multiple myeloma. Prostate cancer specifically, if it gets in the bone, is blastic, meaning that it builds the bone up. So you're going to have this large growth over time, and as the bone is just getting built up and up and up. Multiple myeloma is going to be an example of a lytic osteoclastic bone disease that's going to break the bone down. Many different things can cause cancer, different types of genes, different types of environmental carcinogens, and many different types of pathogens and microbes. Highly recommend this book, again, for anybody who's interested in oncology. He talks about the history of this. We had these three competing groups. It's kind of funny. One group said, we think genes cause cancer. One group says carcinogens, the environment causes cancer. And another group says it's viruses and bacteria and things like that cause cancer. And it turns out not a lot of people really thought maybe we're all right. Maybe it's a little bit of some hybrid of these three theories. Specifically, though, with the idea that it could have been a pathogen, a lot of people thought that cancer was really just caused by a virus. And so if you had cancer, it was contagious, and there was a big stigma, and you were contagious, and you had to be quarantined, and that was just awful, bad, and also wrong. But it actually turns out for Tasmanian devils, there's a type of cancer that they can bite one another, and they spread this cancer from one devil to the next devil. Couple different types of, many different types of environmental carcinogens. Aflatoxin coming from grains or nuts can cause liver cancer. This makes sense because it's metabolized by the liver when you eat food. That seems reasonable. Alkylating agents, so different chemotherapy drugs like cisplatin. This can cause leukemia and lymphoma, especially AML. We'll talk more about this later. Aromatic molecules are aromatic like the ACTG of DNA. They're going to intercalate and screw up DNA replication and transcription. Textile dyes, cigarette smokes, polyaromatic hydrocarbons. So when you're grilling and you get that nice black, crusty char that tastes delicious, that's actually very bad for you. This is going to be especially destructive for the bladder and the lungs. Arsenic, so this is going to be an herbicide. And actually, they used to, a woman's faces are fortunate, it's so funny. You would put this on your face and it would lighten your skin, and lighter skin was considered attractive back then, and it turns out this actually caused cancer. So very bad to rub arsenic on your skin. Asbestos, people have heard about asbestos causing mesothelioma. We all remember those commercials. But it's actually much more likely that asbestos causes bronchogenic carcinoma. The reason that you usually associate it with mesothelioma is because mesothelioma is like very, very... really the only thing that causes mesothelioma is asbestos. But bronchogenic carcinoma could be caused by asbestos or smoking or many other things. Cigarette smoke, we all know lung cancer. Upper GI, that also should make sense, right? It's going through your pharynx, it's going through your mouth, it's going to interact with the cells there and could cause cancer. But bladder is kind of random, but it's definitely an important one to know, is that cigarettes can cause bladder cancer. Ethanol is a similar thing. It's going to upper GIs, we're going to interact with ethanol. But then also your liver. Your liver has to work really hard to detoxify alcohol, and that could cause cancer. Radiation. So radioactive iodine is a natural byproduct of nuclear decay. And so this can mimic natural healthy iodine, get into your thyroid. T3 and T4, those hormones, use 3 and 4 iodines respectively, so it makes sense that it would get trapped there. It can cause thyroid cancer. Nitrosamines are in smoked foods. This can cause GI cancer. And then radon is just a natural, we have uranium in the ground, and this naturally decays, and this can accumulate in basement. So if you get a new house, you have to check the basement for radon. Also, if you guys have watched Chernobyl, you'll see that once they realized there was radioactive fallout from the nuclear disaster, people took iodine pills. The idea is that this would act as a competitive antagonist for the proteins in the thyroid that bind onto the iodine. So a bunch of different genes. BCR-ABL is probably the most important one. This is a fusion receptor tyrosine kinase. So BCR is an upregulated gene in hematopoietic cells. ABL is the receptor tyrosine kinase. And so we have this 922 translocation, which just causes this receptor tyrosine kinase to be way more active than it normally is. So CML, chronic myeloid leukemia. This is mostly associated with the Philadelphia chromosome, BCR-ABL. It's actually also a little bit in the adult phenotype for ALL. Not as common, but we'll see that there as well. BCL2, again, BCL stands for B-cell lymphoma. And I think of the L2, there's an L here and there's an L there. And so this is the lymphoma that's associated with BCL2. Also follicular, there's 2 L's, so that one as well. And again, this is an anti-apoptosis gene. c-KIT, cytokine receptor, this is GI cancer. c-MYC, MYC is a transcription factor. It's associated with Burkitt lymphoma. I always thought of just BC. N, neuroblastoma N, MYC is the same thing. HER2, ERB2, this one's especially important to know. This is a receptor tyrosine kinase upregulated in breast cancer, causes extra growth. JAK2 is a tyrosine kinase a little bit downstream of an RTK. This is associated specifically with chronic myeloproliferative disorders. There are 4 of them we'll talk about, so it's associated with 3 of them. The 4th one is actually just chronic myeloid, CML, but we're just, that one's not associated with JAK, so just these 3. Rett, there's a whole host of cancers under the Rett syndrome, specifically thyroid, parathyroid, and pheochromocytoma. So, in other words, people who have this Rett mutation, this proto-oncogene has been converted to an oncogene, they have too much cell division, they are particularly susceptible to thyroid, parathyroid, and pheochromocytoma. This is a receptor tyrosine kinase that is now overactive. Tons of cancers have BRAF and KRAS, GTPase, serine threonine kinase, respectively. Tumor suppressors, APC negatively regulates the beta-catenin pathway. Is it the one that's associated with Wnt, the Wnt pathway? And familial adenomatous polyposis is a type of colorectal cancer that is particularly associated with APC. BRCA, most of us have heard that breast, and then CA, cancer. BRCA is associated with breast cancer. It's also associated with ovarian cancer. I have specifically this is associated with Ashkenazi Jews as well. I have a Jewish family friend whose mom just got cancer and she was BRCA positive. So this is double-strand DNA repair protein. And CDKN2, then this is just the gene name for the protein P16, which we see over here. P16 is trying to inhibit CDK4 and 6, which pairs up with cyclin D1. This phosphorylates retinoblastoma, which is normally inhibiting E2F. E2F is a transcription factor, which is promitotic. So now that we phosphorylate it, this is gone. This has now been turned off. When it's phosphorylated, E2F can now do its thing, and its thing is to push us from G1 into S. DCC is deleted in colorectal cancer. Go figure, it's colon cancer. SMAD4, so again, this is kind of a similar thing. DPC is the gene name, the protein is SMAD4. This is deleted in pancreatic cancer. Of course, pancreatic cancer. MEN1 associated with MEN2. So MEN1 is going to be another cancer syndrome. So if you have a mutation in this tumor suppressor, you are susceptible to pituitary, pancreatic, and parathyroid related cancers. P10, so P for prostate, N for endometrial cancers, and T for breast. This downregulates AKT and PI3K. Retinoblastoma, RB, this is literally the retinoblastoma protein is associated with retinoblastoma, cancer of the eye, also associated with bone cancer, which is kind of interesting. So as we saw over here, this guy's job is to inhibit E2F. P53 is the guardian of the genome. This is like the most highly cited protein in all of biology. So it's just a major tumor suppressor, has a lot to do with DNA repair. And if you have a knockout, you have Li-Fraumeni syndrome, which is where you get cancers just in tons of different places all over your body. And then the Wilms tumor protein 1 is a developmental transcription factor. This is associated with juvenile nephroblastoma. Cool. Oncogenic pathogens, EBV, Epstein-Barr virus, Hodgkin's, and Burkitt lymphoma. Hepatitis B, hepatitis C, these are associated with liver cancer. This makes sense. This virus goes ahead and infiltrates the liver. Herpes simplex virus 8 is associated with Kaposi sarcoma, largely in people who have HIV that has progressed into the AIDS phenotype. HPV There's many different subtypes, 16 and 18. We all know this is associated with cervical cancer. We've seen the commercials. We know you're supposed to get screened. Less known is that this is actually on the rise over the past maybe 2 decades. Esophageal and oral cancers have gone up, 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 especially in men who are HPV 16 and 18 positive. And there's no polite way to say this. This is due, most people think, due to changes in sexual practices. So oral sex of a woman who has an HPV 16 and 18 just infected cells in her cervix, this can then transmit to the esophagus and lead to esophageal cancer down the road. H. pylori is associated with GI cancer, specifically mucus associated lymphoid tissue lymphoma. HTLV stands for T cell lymphoma virus. Clonorchis is associated with cholangiocarcinoma, so cancer of the bile ducts. Schistosoma is associated with bladder cancer. Grade versus stage. Grade is really just talking about the histological appearance, while stage is much more complex. Stage is integrating many different things, including the grade. So things like the clinical impression, radiology scans, size, did it spread to lymph nodes, did we do a surgical biopsy, et cetera, et cetera. What did the stain look like? This is used for survival curves. This is also used to determine what treatment plan, right? Should we do surgery? Should we do surgery and chemotherapy? Should we do radiation? Should we do them at the same time? Should we do one before the other one? What type of chemotherapy, et cetera, et cetera. So grade 1 is the good one. All of these cells look normal. Grade 3 would be the bad one. And in this case, so here we see the cells are uniform, small nuclei. Here the cells have larger nuclei, and there's a lot of variation in the size and shape. This is grade 1, and then this bottom one here, this is grade 2, and you can tell the differences. So look at all of those dark nuclei that we see. 2 different types of staging. There's the T&M staging system, and then there's the numerical staging system. So, you might hear that someone has a stage, she's got stage 4 cancer. Well, that's the numerical. T&M is a little bit less colloquial. That's the one that you'll probably hear more now in med school and as physicians. There's many different types of T and M. So T is talking about the size, N is talking about the lymph nodes that they've traveled to, and M is talking about whether it's metastasized or has not metastasized. So here notice we have so many different types of ways of categorizing T, so many different ways of categorizing the number of lymph nodes, right? Here we're saying is a carcinoma in situ. Did it get through the muscularis propria? Did it get into the subepithelium? Did it metastasize or not? Notice that this is for a very particular type of cancer. If you have a different type of cancer, there's actually so many different types of TNM staging. So TNM staging for breast cancer is going to be different than the TNM staging for colon cancer, is different than the TNM staging for, Hodgkin's lymphoma, etc. Here's another example. This is a completely different type of TNM staging. You're not responsible for knowing this, but maybe just know that there are many different ways to categorize the TNM. And yet another one. So maybe worth knowing is that the TNM is largely going to go ahead and influence the numerical stage. So here we're looking at N, there's no lymph nodes for this N0 for the TNM staging. And so this is only going to be associated with stage 1A or 1B or stage 2, but it's not in the lymph nodes. It can't be that bad as the idea. And, then if it gets into a little bit of the lymph nodes, then it might be stage 2 or it could be stage 3. Notice all of these are M-not. These are not metastasized. The second you have a metastasis, though, usually you'll say it's stage 4. So some good rules of thumb. Stage 0 is in situ. Stage 1 is it spread to the local nearby lymph nodes. It's local and small. Some people might say it's lymph nodes that are local and nearby, and some people might say it's not at all. It depends on the staging system. But stage 2 and 3 are going to be the larger and more lymph nodes. Stage 4 is specifically metastasis. And again, these are just rules of thumb, many different types of staging for different types of cancer. So it's going to be complex. But most importantly, probably is stage 0 and stage 4. All right, we do have different types of staging. We have a clinical stage. This is the one that's always done. And this is pretty good. I mean, this works. So this is, a physical exam and maybe, a surgical biopsy and some staining, some blood test. They got a CT scan or an MRI, and then the radiologist looks at the size and sees if it's in lymph nodes, and that's the clinical stage. you should always have that if somebody has a cancer. The pathological is specifically if they cut out the tumor and they actually, it's one thing to have a CT scan and say, it looks like 4 centimeters in this direction and 3.8 centimeters in the other dimension. But once you actually cut it out and get, cut out all the lymph nodes, then you have a better idea. So this might add a little bit of precision. The staging that somebody has is for when they first came in. So when you first come in, that is the stage. If you come in and I say it's a stage 2 cancer, and then for the next 6 months I give you chemotherapy, and then maybe it gets smaller, I still say it's stage 2 cancer. And then, maybe it goes away completely, and then it comes back a year later, and you have you're in remission, and then there's a failure, the cancer comes back. We still say it's stage 2 cancer. That's because the treatment options and the chances of recovery they've just found from clinical studies is that it's most highly correlated with how early your cancer is found and the stage that it was in initially. Now, there are exceptions. Sometimes you restage, but that is pretty rare. So as I mentioned earlier, the staging is going to help establish Kaplan-Meier survival curves. Obviously, stage 1 and 2 are going to be the better survival curves, and then stage 3 and 4 are going to be the lower survival curves. Also, the types of treatment. Again, everything is different. Cancers have different staging, different treatments, and et cetera. But we're just looking at cervical cancer. If it's stage 1, it's local. It hasn't gone to nearby lymph nodes, and it's small. So you can probably just hysterectomy, a larger hysterectomy, maybe. If it's stage 2 or 3, maybe you'll give a little bit of chemotherapy. Maybe you'll do radiation. Maybe you won't. If it's stage 4, maybe you'll up the dosage of the radiation, et cetera, or use a stronger dose, more cycles, higher dosage of chemotherapy, et cetera. Different stains that we can use to see what proteins are expressed. An example would be for cancer. So forget this. So this is breast cancer, and you could have breast cancer that's triple negative. You could have it be estrogen receptor positive and progesterone receptor positive, Herceptin 2 positive, this RTK that we talked about. It could be a triple positive, and this is what a normal would look like. And this will just tell us what types of targets we have for this particular type of breast cancer. So that's very useful. So a couple stains to know. If you see the granin or synapto, well, at the end of a neuron or a neuroendocrine cell, you have granules where you'll release your neurotransmitters or your endocrine hormones. And then a synapse is the end of a neuron. So both of these should make sense that these are associated with neuroendocrine cells. Small cell carcinoma of the lung is highly, highly neuroendocrine based. Keratin, we know, is epithelial cells. M for muscle. GFAP, so you're going to see this a lot, especially in the neuro section. G is for glial, and then protein. So these are going to be all of the accessory supporting cells that nurse the neurons. Things like astrocytes and other glial cells, but there can be a few others as well. You probably, if you get a brain tumor that's primary, it's unlikely to be a neuron, because remember, the neurons don't really divide that much, but the accessory glial cells might divide a lot. Neurofilament is a neurons, go figure. Again, this is probably going to be rare, or there might be other circumstances that would explain why the neuron that's usually in a quiescent state is dividing. PSA, prostate-specific antigen, is going to be associated with prostate cancer. This one is really important. Again, not only because the incidence of prostate cancer in men is number 1 and the mortality is number 2, but because it's just a very specific antigen that is produced in high abundance and it's easy to detect. So prostate cancer is very preventable just if you get your PSA checked. So S100 is going to be in neural crest cells. So this is going to be the ectoderm that is in the peripheral nervous system. So your skin and your peripheral nerves. So this would be a melanoma or Schwann cells, we can remember, are specifically in, melanoma is obviously skin, and then the Schwann cell wraps the neurons in the peripheral nervous system. Oligodendrocytes wrap the neurons in the central nervous system. So both of these are peripheral and therefore neural crest. Langerhans cells are the dendritic cells that we see in the skin. TRAP is specifically going to be hairy cell leukemia, which we'll talk about. And then Vimentin, ME for mesenchymal tissue, which we already talked about, which is associated with sarcomas. Sometimes you see these concentric circles of dystrophic, so just like kind of like necrotic tissue calcification. We can get rid of this and get rid of this. So P for papillary carcinoma of the thyroid, S for somatostatinoma, meningioma, mesothelioma, ovarian adenocarcinoma, and then milk, specifically to say that this is a prolactinoma and the anterior pituitary. And these are really big giveaways on histological analysis. A couple of tumor markers. So these are usually very sensitive for a particular cancer, but not specific. That is to say that many different things might produce something like ALP or alpha fetoprotein or HCG. So we don't usually use it as a primary way for the diagnosis. But we can buttress a diagnosis. For the most part, though, we're going to use it to monitor how well you're responding to therapy and to see if the cancer is coming back or how well you're doing. Alkaline phosphatase is associated with phagocytes. So a lytic bone cancer, osteoclasts, remember, have a phagocytic hematopoietic precursor lineage. Liver cancers have Kupffer cells, which are just the name of macrophages in the liver. So both of these cancers are going to be associated with ALP. Alpha fetoprotein, this is a fetal version of albumin. Well, the liver makes albumin, so it kind of makes sense that this particular, in a cancer of the liver, somehow the fetal albumin gene could somehow get turned back on. It's also going to be associated with yolk sac and germ cell tumors, which also would make sense that this is associated with fetal things. Not really related to cancers, but might as well say it. Neural tube defects and Down syndrome are highly associated with alpha fetoprotein. HCG is, this is what the pregnancy tests detect. This is the equivalent to LH. It's secreted by the placenta. It keeps the corpus luteum going, which is the remnant of the follicle. And it's associated with choriocarcinoma. The chorion is really part of the placental tissue, so that one should make sense. Also kind of makes sense that it'd be associated with a lot of germ cell related tumors, so testicular cancer and germ cell tumors. Thyroid produces calcitonin. That one should check out. CEA is carcinoembryonic antigen. So remember that selectins are something that the epithelial will express, which can capture these leukocytes, these neutrophils, can slow them down. They have the rolling, and then they can extravasate through the endothelium and into the tissue where the infection is. And so this particular CEA is largely associated with the selectins in the GI. So it's associated, therefore, with colorectal cancer, which kind of checks out. And one of the effects of this is that it will impair or block neutrophil extravasation and the inflammatory response. It could also allow a cancer particularly to metastasize into or out of. And so we kind of see this here. This is the normal picture, is that they'll bind and then they'll move through. And here, they'll bind, they'll block the neutrophil from binding, and then they will be able to extravasate through. Chromogranin, so we just saw this. Granin, remember, granules for neuroendocrine cells. And this could be many different types of cancer, of the thyroid, the small cell carcinoma, etc. LDH, lactate dehydrogenase, this one's kind of random, germ cells or ovarian tumors. And then PCA we talked about is specific for the prostate. CEA 19-9 is pancreatic, 125 ovarian, and then 15-3 is breast. I wish I had a fun mnemonic for that, but I don't. A bunch of paraneoplastic syndrome. So paraneoplastic syndrome is really just something that a cancer causes that is seemingly distant and unrelated features. So it's just kind of a, for lack of a better way to put it, it's a random thing that the cancer causes that is just a very noteworthy phenotype, a clinical presentation that is just seemingly bizarre. Like if you had a brain cancer, you have a headache. That makes sense because you got a big tumor in your head. But that wouldn't be a paraneoplastic syndrome. That's pretty direct. But something like a GI cancer causing, cancer of the stomach causing acanthosis nigricans, something associated with diabetes and obesity of the axilla, and the folds of the neck, and the sign of lesser trelat, seborrheic keratoses in the back. These are kind of random. So this is random, very, very random. And so that's how I always think of paraneoplastic syndromes. So if someone is hypercalcemic, parathyroid hormone-related protein, many different types of squamous cell carcinomas will produce this, and this will cause calcium levels to go up. Or it could be lymphoma. Lymphoma is, in the lymph, we have a lot of macrophages, and macrophages possess the enzyme 1-alpha hydroxylase, which is necessary to produce a functional vitamin D, which will cause more calcium to be absorbed in the small intestine. Cushing's disease, so too much cortisol. Hyponatremia, because we have too much blood volume or too low sodium count. And then Lambert-Eaton myasthenia. So all of these are going to be small cell lung cancer. So ACTH for Cushing, ADH, antidiuretic hormone, also known as vasopressin, to cause the hyponatremia, and then an antibody for calcium receptors to cause the LEMS. So small cell lung cancer, and I bolded this one because it's important, it comes up a lot, causes a bunch of random phenotypes, and again, that's why this is perineoplastic. So LEMS is very related to myasthenia gravis. So we're talking about a thymoma here. An antibody against the postsynaptic acetylcholine receptors. So the reason these are related, remember that for muscular contraction on the presynaptic side, the action potential moves down, and this causes a depolarization, which causes calcium to flood in, which causes the vesicles to fuse with the synapse, releasing the neurotransmitters. And the neurotransmitter acetylcholine binds to the postsynaptic receptor, which causes a muscular contraction. So whether or not I have an antibody for the calcium receptors or I have an antibody for the postsynaptic acetylcholine receptors, I have a similar phenotype, similar clinical presentation of abnormal muscular contraction. So that's the first thing of the thymoma, the first perineoplastic syndrome. Second would be red blood cell aplasia. I don't actually know what causes that, so I don't have a good answer there. And then finally, because you don't have T cell development, you have a hard time stimulating your B cells, and you have hypogammaglobulinemia, so you have a deficiency of antibodies. Polycythemia is due to excess or secondary polycythemia. We will see polycythemia vera, which is a primary, which is coming later with the myoproliferative disorders. This is due to excess EPO production. Well, it makes sense that a kidney cancer might do this because the kidneys typically produce EPO. Kind of randomly is that pheochromocytoma, a cancer of the adrenal medulla, might also cause excess EPO, which could cause polycythemia. Tumor lysis syndrome. So yes, cancer cells are dividing a lot, but they're also dying a lot. And when they die, they spill a lot of their guts into the blood. Things like potassium, things like phosphate, a bunch of nucleotides. So the nucleotides, there's so much, remember we see this high nuclear to cytoplasmic ratio that we don't see in normally cells. So there's so much DNA and not a lot of stuff in the cytoplasm. So much DNA, so when the cell dies, you lose a lot of ACs, Ts, and Gs, which means you get a lot of phosphates, which also means you're going to have a lot of your nucleotides broken down into uric acid. So the phosphate specifically will also bind into the blood to calcium. So the blood calcium levels go down. Nucleotides are broken down into uric acid. So the phosphate, calcium crystals, or I guess more just the phosphate here is causing the crystals with uric acid, can cause kidney injury. Low calcium levels can cause seizures and tetany. Changes in your ECG. High potassium levels can cause arrhythmias and muscular weakness. Okay, cool. Hope that was helpful. Part 2 coming up.